So here's a brief outline of what I want to cover in the next sort of 20 minutes to half an hour. Just go through some of the basic anatomy and cover some of the points that seemed most relevant for this application. So here's a diagram of the basic anatomy of the eye. So we have the cornea, the horn-like substance across the front, then the lens in the middle. I'm just highlighting the kind of most important structures that I'm going to talk about. The lens and the cornea together focus the image on the back of the eye, the retina. We're going to talk about the fovea, where the central region where vision is most acute. And the optic nerve, which is like the cable taking information to the brain. I'm only going to very briefly talk about the brain. I'm not going to cover the anatomy or anything in any detail, but it's worth just pointing out the key visual pathways. So the optic nerve, as I say, take information to the brain, ultimately to primary visual cortex, which is at the back of the head, and then on to other visual areas. And you can see that the optic nerve actually crosses over at the optic chiasm, so half the information from each eye goes to each half of the head, if you see what I mean. And the effect that that has is for information on the right side of the visual field is processed by the left half of the visual cortex, and vice versa. The key features of the eye that I want to talk about are laid out there: the lens, pupil, retina, and optic nerve. So people sometimes think that the lens is what focuses the image on the retina, but actually it's almost entirely the cornea. So the cornea has a power of about 40 diopters, and the lens has only about 20 when it's relaxed, and then some ability to increase beyond that. So what the lens does is give us our ability to vary the power of the optics of our eye. And as you can see, they both have quite a high refractive index, comparable to that of glass. Now, the ability to accommodate the eye is something that changes with age rather dramatically, as you see here. So those of us who are getting past 40 actually have very little ability left to accommodate, hence the reading glasses that we increasingly need to read. And I think that's something that's maybe not always appreciated enough, that because, as we'll see in conventional stereoscopic displays, accommodation is a particular issue. It's something that's likely to be very different for young people and old people watching the same displays. Older people have little ability to accommodate anyway. So I thought it was also worth talking about common refractive errors that you encounter in the human eye. People like me are nearsighted, have an eye that is too long for the power of our lens and cornea, so that the image is actually brought to a focus in front of the retina and therefore appears blurred on the retina. And that can be corrected by putting a diverging lens in front of the eye, whereas those who are farsighted have the opposite problem, that the image is naturally brought to a focus behind the retina. Something I thought I would just mention, because it does come up surprisingly often, of course the image that's formed on the retina is actually upside down, and surprisingly often people seem to think that's some kind of a problem or a challenge for understanding vision. But of course it isn't, unless you're thinking that there's some little man in your head that's looking at the images on the back of your eye. All that's important is that your brain knows the correct mapping from the image on the retina to the outside world. So it's no more important that the image is the right way up on your retina than it would be that an image of a photo stored on your computer should kind of be the right way up on the hard disk. It's really not a question that makes sense. Now, the pupil, as we all know, has this ability to constrict and dilate to control light entry, and that's one of several mechanisms that the human visual system has to enable us to operate over a huge dynamic range, a huge range of light levels. And actually the pupil is a relatively small component of this adaptation. Most of it is neural, most of it is a shift in the sensitivity of our photoreceptors. But the pupil does also constrict and dilate, and of course that affects the depth of field. So I guess under optimal circumstances you have a lot of light and the pupil can constrict, so it becomes much more like a pinhole, giving you a wide depth of field and ensuring that objects at many different distances are still in focus. Whereas at low light levels the pupil expands in order to catch all the photons that are available, and obviously that gives us a much narrower depth of field. there's actually a relationship between convergence and accommodation and pupil dilation. So the pupil has this tendency automatically to constrict on near viewing, independent of the light level, possibly again to try and keep the object more in focus there. Interestingly, pupil dilation also, independent of light, can also indicate arousal and attention. So there's a whole field of psychology called pupilometry, where you measure these fluctuations in pupil size in order to get an objective measure of attention and arousal. And that's been used historically. The deadly nightshade, as it's known in English, this plant Atropa belladonna, beautiful lady, it contains a poison which actually dilates the pupils and is used clinically, atropine, for that purpose today. But the name belladonna came because apparently hundreds of years ago, women would put little drops of this into their eyes to make their pupils expand and make themselves appear more attractive and I guess more interested. Moving on then to the back of the eye, the retina. So obviously this is critical. These are the sensors that record the light falling on the eye. Kind of interestingly, vertebrate eyes are kind of inside out. So the light has to travel through all the neural machinery before it reaches the light detectors, which are at the very back of the eye. And this kind of seems an odd way to design a system. And some species have evolved differently. So invertebrates like squids have their light sensors on the inside of their retina. So they're the first things that the light encounters. And you'd have to say that seems a better design. But for whatever reason, we evolved this way. So the light receptors are at the back of the eye. And then we have quite a lot of fairly complicated retinal machinery that actually does a lot of image processing before the image even leaves the eye. And the output layer are what are called the retinal ganglion cells, and it's their axons, their nerve fibres that form the optic nerve and carry information to the brain. And the only information that leaves the retina is along these nerve fibres. Talking about the photoreceptors, as I'm sure you all know, humans have 4 classes of photoreceptors. We have one rod type of photoreceptor and 3 cones. But the rods only operate in very low light levels. They rapidly become saturated. And the cones are fairly insensitive, so they only operate in high light levels. So effectively, in low levels we have scotopic vision, adapted for darkness, and we're then monochromat, so that's why we can't see colours at night. Whereas in high light levels, so daylight for example, we have 3 types of photoreceptor operating, so we have trichromatic vision. And so, of course, it's the cones that give us our colour vision, but actually we're relatively insensitive at picking out the spectral information. The entire spectrum of light is reduced to just 3 numbers, which is obviously a pretty crude representation. And again, other species do better. Many birds, for example, are tetrachromats, so they have 4 kind of cones. But I guess from an representing visual reality kind of way, it's rather handy that we have this small sensitivity to spectral information, because of course it means that we only need 3 primaries in principle to reproduce almost the entire colour gamut. This is an image of a slice through the retina, through where the optic nerve leaves the retina. So I thought this was a beautiful picture. You can see all the nerve fibres from the retinal ganglion cells streaming out of the retina at the optic nerve at the point where it's plugged in. And there, of course, we're blind because we have no photoreceptors at that point. They've had to be cleared away to make room for the fibres to move out. And it's kind of interesting to think that we each have these 2 scotomata, these 2 blind spots, one in each eye, that we are absolutely not aware of in everyday life. Partly that's, of course, because the blind spot in one eye, we can generally see the objects that fall in our blind spot in one eye can be seen by the other eye. But it's not just that. I mean, I close one eye, I'm still not aware of having a blind spot because my brain automatically fills in the missing information in quite a clever and quite an active way. The other thing I wanted to point out about the optic nerve is that the retina has an enormous amount of compression. So we have, estimates vary, but around 10 to the 8, around 100 million photoreceptors, so something like 7 million cones, and actually a far greater number of rods. But we only have about 1 million fibres that leave each eye. And you can see that's an absolutely massive compression, around 100-fold. So already at the very early stages of vision, the processing in the retina has thrown away most of the information that was actually collected by the photoreceptors. And that only happens, you know, again and again in the brain as attention and so on zooms in on what we're going to notice. So that's another very characteristic fact of human vision and accounts for a lot of the mistakes, if you like, that human vision can make. This is another image across the retina. So this is showing the fundus, the back of the eye. Here's the blind spot. Here in the middle is the fovea. The macula is the word for the region around the fovea, and the fovea is the little pit-like depression right at the centre. So you can see how specialised the fovea is. That all the support mechanism of the retina, the blood supply and so on, has kind of been moved away in order to enable light to have the best access to photoreceptors right at that point. And the fovea is absolutely critical for vision, for our visual experience. As you can see here, this is how specialised the fovea is. So what's plotted here is photoreceptor density for the cones and the rods. And you can see that the cones are absolutely crammed into this tiny, tiny area of the retina. And we have very few actually beyond about 10 degrees eccentricity in our visual field. The rods, on the other hand, I mentioned we have many more rods than cones. And you can see why here, that obviously most of the retina is outside the fovea, and that's where the rods are. So they are much more important for our peripheral vision. This is a beautiful image of the cone mosaic around the fovea, and it's colour-coded according to the spectral sensitivity of the cones, so the red, green and blue, basically long, medium and short wavelength sensitivity. And you can see that right at the centre of the fovea, the cones are even more densely packed than elsewhere. And you'll also notice that the red and the green, they're only red and green photoreceptors right at the very centre. So even just a degree or so away from the centre of our vision, so within that very central region, we don't actually have any blue photoreceptors. So our colour vision's even more specialised just right at the very centre. Something I didn't mention, but it's maybe worth throwing in there. You'll have noticed a few slides back when I plotted spectral sensitivity that the red and the green cones have got rather similar sensitivity, and then the blue cones kind of out on the side. And the reason for that is that red and green actually mutated off from one another relatively recently in our evolutionary history. So we used to have two cone types, long and short wavelengths, and then the two long ones split off into long and medium. And we can still see remnants of that in how they're processed in the brain. So the consequence of the specialisation of the fovea, the extreme density of cones at the middle and the relatively lack of cones outside the fovea, is that we actually see well only in a very narrow region around the fovea. And again, because our brain works so cleverly, we're rarely aware of this. So as I stand and look at you in this auditorium, I have the sensation that I can see the entire room in perfect detail. But in fact, I can't. I only see it at all well around 3 degrees, like holding up my thumbnail at arm's length. That is the extent of my high acuity vision. And around that, I don't have reliable information. Now, I really like this image, which is from a fairly recent paper, which I do recommend you read if you're interested, because I really like that paper. And what they've tried to do here is give an image, or give us an understanding of what our vision does in the periphery. Now, earlier versions of this image would have represented this peripheral area as blurred. It's like you've got very low acuity there, so an obvious way to represent that is like a blurry image. But actually, that's not how we see, and we can tell the difference between an image that's blurred in the periphery and a correct, the veridical image. Rather, it's kind of things get scrambled. So the idea is that these are called metamers. A metamer is a word meaning things that you can't tell the difference between. Okay, so you might have come across that in the context of colour. We can't tell the difference between a single wavelength orange light and a combination of an appropriate mixture of red and green. They both look orange to us, and we can't tell the difference. They're metamers. Now, these images are metamers in the sense that if you fixate here, and then I flash up, either of these two images, they both look the same to you. You can't tell the difference because in the centre where you're looking, they are exactly the same, and this one only starts to become different in the periphery. If you flashed up an image that was blurred in the periphery, even in a brief presentation before you had a chance to move your eyes around it, it would look different. You'd be able to see that it wasn't the same. So yeah, that's how our brain is representing the visual information on the retina. So the consequence of that is if we want to see something well, we have to point our fovea at what we're interested in. And that's why the lecture theatre looks uniformly sharp to me, because in fact I'm flicking my eyes around it all the time, making these saccades jumps about 2 to 3 times every second without any kind of conscious awareness. And we have this amazing system of eye muscles which are beautifully engineered to give these really rapid jumps and moving like 200 degrees a second, but incredibly briefly, and then coming to a complete stop. So it is a beautifully engineered system. And I've got a demo here, actually, of just how important it is to be foveating the object of interest. So here's a scene. It's a GIF animation. And I'm sorry it's a bit annoying and jumpy like that. But what's happening is that actually the scene is being flipped back and forth between 2 different images. And can you put your hand up if you've seen the difference between those images? I've got 1 hand. 2. A few more coming up now. Yeah, but still quite a lot of people who've not seen the difference, I think. So what you're having to do, of course, you will only see the difference if you happen to be foveating the point where it occurs as the images jump between them. You won't notice it if it's in your periphery, precisely because the information in your periphery is scrambled. Anyway, has everyone seen it yet, or are there still some people who are... No? Shall I tell you? So if I put the cursor here, you foveate that.
Can't see the cursor there.
Oh, sorry. I thought it was mirrored on that screen. It's the bar behind the couple. But it's rather hard to notice because your brain analyses a scene. We're humans, we're interested in the people, we're following their gaze. The bar, it's kind of a large and obvious structure in the scene, but it's not one that's very socially important to humans, so it's not something you would naturally foveate in that scene. And unless you foveate it, you won't notice the jump. Now, obviously, the reason we have to put the grey screen in between is that humans are very sensitive to motion within an image. So if I just put the images jumping back and forth without a blank in between, of course it would be immediately obvious to all of us because it would be the only motion in the scene. But the grey screen just serves to kind of wipe everything. And then it just reveals how poor we are actually at how little information we consciously perceive about a retinal image. Okay. Now, eye movements, obviously, as I've just laid out, they're important for all vision, but they're actually particularly important, or arguably particularly important, for binocular vision. We have 2 eyes, and we need to point both of them at the object of interest. And movements of the eyes in opposite directions in the 2 eyes are called Vergence movements. The idea is if you want to look at this apple, then you need to bring both foveas to point at it. So we have this terminology that version movements refer to parallel movements of the eyes to look in a particular direction, but vergence eye movements are there to try and reduce the disparity of the object on the retina. By disparity, I mean the difference in the retinal location at which it falls. So obviously, if an image is falling at the fovea in both eyes, it has 0 retinal disparity. So when you're fixating this red apple, it's got 0 retinal disparity. The green apple is at a different depth, and it projects actually to different positions in the 2 eyes. So you can see that the angle of this, the difference from the fovea in this eye is much smaller than in this eye. And that's what we mean by saying that this green apple has got a retinal disparity. And in this case, a crossed disparity, because lines of sight from the 2 eyes cross, intersect here in front of the fixation point. Oh, I haven't distributed I brought some 3D glasses to show the low-tech form of visual displays. Oh, sorry, here they are. Yeah, sure. I mean, it's only this one image really, so it's possibly not worth it. And I guess we've all seen these kind of 3D displays. I'll come back to that image in a bit when you've handed it out. But that's the basis of stereoscopic displays. So stereoscopic 3D uses some form of glasses or special filters or maybe an auto-stereo display. Somehow or another, it presents different images to the two eyes. And then, of course, you can simulate an object in space. And it's a kind of interesting property of human vision that this retinal disparity is sufficient to give us a perceptive depth. It's not necessary, but it suffices on its own. So we can give an impression that the object's floating there in front of the screen. I also wanted to, just as a matter of terminology, sorry, I don't know why they're auto-advancing, just point out that it can be handy to discriminate, to reserve the term parallax for distance on the screen. So I like to keep parallax to mean the separation between the two images on the screen and reserve the term disparity for what happens on the retina. And there's no unique mapping between screen parallax and retinal disparity, okay? So if the viewer turns their eyes to fixate this object in front of the screen, then its image will have zero disparity on the retina because it will be falling on the fovea in both eyes. And it will actually be this image here, which has no screen parallax, that will have a retinal disparity. Is that making sense to everyone? Great. Now, it's an exciting time, I think, to be a 3D vision researcher because there are all these new technologies coming out. So Parallax Barrier is one it's quite an old-fashioned technique. I believe it was at the beginning of the 20th century that people first started playing around with parallax barrier displays, but they've definitely had something of a renaissance. And so that's a way of presenting different images to the left and right eyes without the need for wearing special glasses. And I'm excited in these sort of developments and always got an eye out for how they can be applied within my field. And I brought an example with me here, actually, of a 3D tablet that my lab is using to develop a vision test for children to be used in eye clinics and help with the diagnosis and management of binocular vision disorders. And we think that because we've now got a 3D tablet, we can embed the vision test in a game and kind of make it fun and engaging for children in a way that the existing clinical tests really aren't. They're quite boring and kids often don't want to do them. Yeah, so if you want to have a look at that afterwards, I'll be happy to pass that around. And I guess that's one of my motivations for being here, really, and for finding out about new technologies that are coming along for immersive visual displays, is that these have applications in all sorts of areas, including medicine. Okay, so more about eye movements. So screen parallax in the object that the viewer is likely to be interested in generally stimulates eye movements which are designed to null out the retinal disparity. And the reason for that is that we can't fuse large retinal disparities. So our visual system is only wired up for really quite subtle differences between the two eyes' images. And if you present bigger disparities than that, we won't be able to fuse them and the image will appear double and it'll be very disturbing. So as we've seen, you need to converge the eyes so that whatever you're interested in projects ideally to the fovea in both eyes. And of course, that means you also need to accommodate, you do also need to accommodate so that the object will also be sharp and in focus on both eyes. And that depends on the light level, of course. It's particularly important at lower light levels where the pupil's likely to be greater. So you're accommodating by making this lens thicker and therefore more optically powerful. Now that's a bit of a problem with the conventional two-view stereoscopic 3D displays. So in real life, you've got this lawful relationship between where you need to focus your eyes, near or far, and where you need to converge your eyes. Okay? They always agree because you're looking at a real object that's at a particular depth. But on a 3D display, a conventional stereoscopic 3D display, the images are in fact on a cinema or TV screen at a fixed focal distance. But you still need to converge or diverge your eyes to keep the object on your fovea. And so you've got this mismatch between where you need to focus your eyes and where you need to point your eyes. Highlighted there. And it's known as a Vergence and Accommodation conflict. And it's a problem for the following reason. Because vergence and accommodation usually go together in natural imaging, in natural views, we've evolved mechanisms, we've evolved links between them. So they're neurally linked in the brain. So retinal disparity on its own, of course it triggers vergence, as we've seen, and blur triggers accommodation. So if I perceive that this object is out of focus, then my visual system will automatically try and adjust the lens to bring it into focus. But also, because they always go together in the real world, accommodation triggers vergence movements, even on its own. So even if you occlude one eye, as here, and then this eye that's not occluded views an object that's close up, that will trigger a vergence movement that is at least roughly appropriate for the near viewing distance. And similarly, if you are converging on an object in front of the screen, then, oops, it's advancing automatically. If you're converging on this object because you want to look at it and it's in front of the screen, you will have a tendency, therefore, to accommodate appropriately for an object at that distance. But in fact, of course, the object's on the screen, so you actually, if you want to keep it in focus, you have to be accommodating for the screen distance. And so you're always kind of fighting against your neural link between accommodation and vergence when you view a stereoscopic 3D display. Now, it works. 3D displays work because we have a reasonable amount of tolerance to accommodation and vergence error. So the eye's depth of focus is generally quoted as around plus or minus 0.3 diopters. So within this range, the image is going to appear relatively sharp. You're accommodating here, let's say, but on either side, the image is still going to appear sharp. And similarly, we have a range of binocular disparities that we can fuse, 0.07 to about 0.14 diopters. It depends on the stimulus properties and so on, so it's kind of hard to give a precise figure. So images with this kind of range of depths are going to appear single. And so you can, if I plot the focal distance against vergence distance for a real object, obviously these two distances are going to be the same, but there's some zone of tolerance in between them where it still looks okay to me. And so if you want to present things on a 3D display, you have to make sure you don't go outside that range. You're going to be fixed at this focal distance because the screen is at a given distance from the observer. So you need to make sure that you don't ask them to verge too far in front or behind the screen. Now, I think it's somewhat of an open question just how much of a problem the vergence accommodation conflict is. People in the field kind of argue about this. I mean, we certainly know that large conflicts unambiguously are a problem. They increase your time to perceive stereo and so on, but probably most importantly, from an application point of view, they give you eye strain and fatigue and headache, and it's kind of, it's just horrible to look at. It's not really clear, I think, whether how much that contributes to the problems people report with viewing 3D cinema. So anyone who works on 3D will know that if you tell people, oh yeah, I work on 3D, they come and say, oh, I hate 3D cinema. Oh, I went to see Avatar in 3D and it was awful and it made me feel sick. You know, I think it's an open question. How much of that is the vergence accommodation conflict and how much is other factors? So the 3D industry is very aware of the vergence accommodation conflict and works very hard to keep Avatar or whatever within this comfort zone. So it should look okay. Is it possible that the residual discrepancy is responsible for the remaining adverse effects? Possibly. I think probably also other factors contribute. So simply the fact that there are conflicts between the visual information that says, you know, I'm flying over the planet Pandora, and the vestibular information that says, no, I'm sitting in a cinema seat. And I think we're used to kind of discounting those conflicts for 2D information because at some level our brain has learned that 2D visual information isn't real on a screen. I can ignore it. It doesn't mean there's a problem. And maybe for 3D, we kind of trust it more. And so our bodies get more upset when there appears to be this massive conflict between two of our senses. Now, full parallax imaging, I'm excited about that because it's definitely going to solve the vergence and accommodation conflict. And then it's going to be interesting to see whether these other problems, dizziness and sickness and so on, still persist because of the other factors, or whether solving the Vergence accommodation conflict really leads to a huge improvement in viewer experience. Anyway, I'm going to leave it there, but please ask any questions and I'll do my best to give answers. Thank you.